Good evening, everybody, and welcome to all our students, alumni, and other friends from around the world who are joining us. My name is Jonathan Mickey. I'm president of the college, Kellogg College, so welcome to everybody, particularly if this is your first visit. My job is to introduce the person who will introduce the speaker. You can always tell how important an event is, whether there is someone to introduce whoever's going to introduce the speaker, because this event is organised jointly with the Oxford International Biomedical Centre, whose president, Charles Hašek, is here, and so he will be explaining the link and introducing the speaker. So Charles.
Okay. Well, I think most of you in this room probably know who Anne McLaren was, outstanding developmental biologist who sadly died exactly 15 years ago in a road accident. And since then, we've been having these lectures, and we're particularly pleased today to be able to welcome Professor Liz Simpson, who is herself a distinguished immunologist working largely on cytotoxic T cells, or has been. Anne McLaren was a person who was very determined to improve the status of women in science, and I think I can honestly say that Elizabeth Simpson has followed that lead in a very large measure, and I presume everybody in this room would agree. So anyway, without more ado, oh yeah, we used to try and get speakers talking about topics as closely related to Anne's work as possible, and we've covered quite a wide range. This year, we're particularly fortunate because Professor Simpson actually worked with Anne McLaren in her very lab, correct? So without further ado, I introduce Professor Elizabeth Simpson, who's going to be talking about Anne McLaren, colleague and friend.
Thank you for those kind words, and thank you for the honour of inviting me to give the Anne McLaren lecture in, I was going to say 1922, but I should say 2022. Yes, Anne and I were colleagues. We were in different labs, but we used to meet up. We were both labs that were in London, both MRC labs, but we came together to do experiments, and we nipped to and fro each other's labs, often with pieces of mice in test tubes taken from mice in one lab to be analysed in the other. However, let me cut to the chase and continue along these lines. We actually met at a party, rather appropriate because Anne and I both like parties. It's a wonderful way of meeting people. And it was a party in Peter Medaware's house in Christmas time. And it was just after I'd given birth to my daughter. I think she was a few weeks old at the time. So of course I took her with me, and clearly approved of that. Her children had also been taken with her to various events from the time they were small. And there she is, a photograph taken in the year that she was elected FRS, Fellow of the Royal Society, and indeed the end of that year when I met her. And I'm going to talk about her and her work, her ideas, her work both in science and in the community, which was linked to the work in science, under a number of headings. That of her being a very curious person is the first one, and perhaps the most important. Anne grew up in a family where she wasn't short of material means, but she did not have a conventional education of the sort that her brothers did, to take them to Oxford. She became very interested in ideas and science. Her family were full of interest in a number of things. She had several suffragettes amongst her, in her background. They had interesting people visiting the house, going for walks on the grounds with them, talking over dinner. And that also, I think, fueled her interest in things, ideas, and in making observations and finding out about new things. So she was determined to get an education, and she persuaded her parents to let her to take a course in a grammar school which would fit her to do the entrance exam to Oxford. And in those days, this was in the immediate post-war period, for the Second World War, and there were relatively few, very small proportion of women who went to Oxford, and Lady Margaret Hall clearly were impressed by her potential, and she was offered a place there. But she was very determined to have got there. She had originally decided that she was going to read English, and then she realized that because of her education, she'd missed out on an awful lot of background reading. And zoology looked really interesting, and there were some interesting people in the zoology department. So she opted to do a zoology degree. And that's really when her interest in science took off, with her first degree, but then subsequently doing a DPhil. In that department, she met Medawar, who was doing his experiments on skin grafting at that time. She met Adrian Mitchison, who was JBS Haldane's nephew, and also he was doing a PhD with Medawar on transplantation, and himself went on to be continued to be a very important influence in immunology. She also met Donald Mickey and started to do some experiments with him. And by the time they both qualified with their degrees and postgraduate degree, they were married and had decided to take two posts as postdoctoral fellows in London, in University College London, which now Medawar was a head of department. And the work that they'd started in Oxford had to do with development. And this Anne and Donald took forward with determined strides to look at the factors which influence development and birth of mammalian young, that could be separated into those that were responsible, were due to genes, the inheritance, and those that were influenced by environment. Now, the environment of a fetus is the mother's uterus. And so Anne devised a number of ways of separating these two by taking very early embryos before implantation from the mother mice, and watching them, exposing them to various conditions in vitro before putting them back in the uterus to develop to full term. And these were really precursor experiments for in vitro fertilization. There was a huge amount of work to be done on that. The actual conditions, when you could do things, the medium that you needed to use, how you prepared the uterus of the mother who had the uterus that you were wanting to implant the new embryos into, the manipulated embryos, all of that had to be worked out. And although the conditions that she established were obviously relevant to mice, an extraordinary number of the basic principles were exactly the same for other mammals, including human beings. You sometimes hear people rubbishing experiments that are done on mice, because humans are so different. Well, they're not that different. During that time, Anne had 3 children, and when she was ready to move to a new post in Edinburgh, they went with her, and she looked after them, very often taking them to the laboratory with her, and allowing them to go into the animal house, having cages of mice which were there, And I really perhaps should ask the children a little bit more detail about that. But Anne was insistent that my daughter, Emma, as she grew up, should have access to experimental mice. And whereas in my institute, that sort of thing was frowned on, Anne allowed her to come into her animal house. And there was a cage of mice that were kept for my daughter to handle and look at and be interested in. Now, in Edinburgh, Anne focused her work on more mouse development and extended it to chimeras. Chimeras are mosaics of cells from two different individuals that are pushed together very early on in development and together jointly make the animal which is born, the fetus. I'll go into that in a little bit further with the actual experiments. When she moved down to take over she was a founder director of the Mammalian Development Unit at UCL, which was funded by the MRC. She became particularly interested in the development of particular cell lines, particularly those that gave rise to testis in males and ovary in females. So sex determination and what triggered the development of maleness became a very great interest to her. And that was focused on the Y chromosome. As you'll see later, I had a gene, which is a transplantation antigen, that was on the Y chromosome. So that also helped to bring us together. Now, in 1992, she had reached an age when the MRC traditionally retire their directors and close the units. They certainly didn't want to lose Anne, and they offered her to continue funding the unit. But she didn't really want to go on looking after a unit and lots of other people. She wanted to do experiments of her own. She moved down to Cambridge, worked in the Gurdon Institute, had a lot of colleagues who were interested in the same sorts of things that she was. So she again moved and generated more information. But at that time also, she undertook a lot of other activities. And again, I'll come back to them later. Now, these were some of the people that she met both before she went to Oxford, H.G. Wells. And in fact, she took part in one of his early films. So it might have been Anne McLaren, film star, but I think that curiosity about things scientific drew her away from that. But he was certainly an influence and had been a friend of her parents as well. Peter Medawar I've already mentioned, and after the skin grafting work, he went on to work with two colleagues, Leslie Brent and Rupert Billingham, and do some very important experiments on transplantation tolerance. So she was around when all of those ideas were being discussed, when the results came through. And JBS Haldane was in that same department as well. Also in that time, in the early 50s, Donald and Anne, and you can see on that slide there's actually an arrow. This photograph was actually a photograph of a photograph, and I can see that I accidentally put a hand over Anne's, but I don't think that anybody could miss realizing that that was Anne. She's the only woman in that photograph, and Donald is standing beside her. And then in the background, one of those men is in fact the tall one, is Hašek. And I'll tell you a little bit more now in a moment about Hašek's experiments, which I think were an important influence on a number of people, including Anne. But I also wanted to mention the joint experiments that Anne and Donald did together on mouse embryos. And I'm going to do this by showing you the title of a publication that came out in 1958. When they were both working in Medawar's lab, but also some of that work was done at the Royal Veterinary College just down the corner, because they had more space for doing mouse breeding. In these slides from now onwards, I'm going to show you the publications. I'm not going to describe the publications, but just to indicate the sorts of journals that they were, the titles of what they were doing and the sorts of journals they were being published in. As we go through. They're a tiny selection. These are the ones I'm going to show you, a tiny selection of her huge output. This is another selection of her huge output. And here is Donald sitting in the middle, squashed between his two sisters, Susan and Caroline. Anne was, as I've mentioned, very open about moving between the lab and home. I have no first-hand experience of that, and I think I ought to get myself into conversation with Donald to check out more of these details, which would be of great interest to me. Now, to go back to the question of Anne's research on embryos that were grown briefly in tissue culture, a crucial paper that she published with John Biggs, who was also an internationally important scientist in this area, laid the foundation of IVF because it showed that you could explore genetic and environmental influences if you took embryos from one genetic source, replace them into a mouse of a different inbred strain, and then you could evaluate them at birth, count them first, evaluate them at birth, and see what effect it had. Now, Anne went on working on this for a number of years and creating a huge amount of data in an area which was obviously had implications for human medicine, which she was aware of but not pushing. She was interested in the science, and there were people reading her papers at that time who picked that up, and this did lead on. Let me just show you, first of all, though, a page from her notebook. She was absolutely meticulous in her record keeping. Here you can see individual mice being identified with respect to not just their sex, but all sorts of other genetic markers which they were carrying, because this is how the experiments were set up. So notebooks, page after page, notebook after notebook, and this is a very important source of, and when I was working with her too, notebooks were a very important part of what we were doing. Now, back to Hašek and a different species of embryo, chick embryos. The advantage of birds is that their embryos develop in eggs, which are very easily accessible. And here's a couple of eggs that have been put together by Hašek. The shells broken in just over a small area, and the underlying vascular membranes joined together so that they could actually fuse, and the red blood cells from both embryos circulate in each one of them. So they then became a mixture of cells. Each one of these chicks became a mixture of cells of the one that they'd been joined to during development and their own. Now, at that time, Hašek, this work was first published in Czech, in a journal which was probably not widely read in the West, although Peter Medawar and Anne and Donald were aware of this work that was being done. And there were two other people who were working on this. Well, one of them, of course, being Peter Medawar, but there was another American working on this, but with calves. So 3 different species looking at the effect of shared circulation between mice with respect to work of Peter Medawar, chicks, birds with respect to Hašek, although those experiments were also repeated by Medawar, and also in cattle with an American scientist whose name I've dreadfully forgotten for 1 second, but it'll come back to me, so I shall interject it later. Ray Owen. See, they don't take very long to resurface these names. He was a wonderful man, and he was looking at cattle that were co-twins sharing a placenta. So that was not an experiment, that was an analysis of a natural occurrence. And they all ended up by sharing, each of these species, they shared circulations. Medawar was the one who took it to the test to see what was the result of sharing circulations, and found that it made them tolerant of each other's transplantation antigens. So when they were adult, you could actually take skin from either of the two parental strains and put it on the animals that had been chimeric, that were chimeric. Hašek was very fearful of interpreting in this way because the Communist Party would rather that it was a confirmation of Lysenko's ideas. He was a botanist who had actually fraudulently claimed evidence of inheritance of acquired characteristics in plants. Which was a finding much approved of by the authorities in Russia. And in order to keep his nose clean, Hašek sort of went along with that without being too explicit about it. And that way he retained funding for his institute, but gathered a certain amount of ire from other scientists. But he was an extraordinary and very bright man, and Medawar liked him very much. Now, so Anne then worked on, I don't know whether her work on chimeric embryos actually derived from her interaction with Hašek, but the fact that she'd actually worked with embryos in vitro and was in a position to push them together, that may have happened at the same time or been influenced by Hašek's experiments. And she fused embryos from different mouse strains. And here's one paper in a research paper that was published in Nature in '69. But she also wrote a very nice textbook on development of how to make chimeras and how to use them. And that was a very neat little book that came out in '76. She was interested in the interaction between maternal and fetal tissues during pregnancy in mammals. And she therefore was able to use chimeras to investigate the movement during embryonic development of specialized cells as they migrate through the body and take up position where they're going to take part in the formation of the heart, the brain, the gonads. And it was this decision to these cells to move into the gonadal ridges and then subsequently decide to be either testes or ovary that became a pressing piece of curiosity for her. What was the trigger for these undifferentiated cells to turn in one way or the other? The first trigger, in fact, is testis determination. It's not the only one, there's a series of it, but the first trigger is one that she wanted to pin down. But she was also interested in the possibility that the differences between fetuses that carried their father's transplantation antigens, if he was a different strain of mouse or a different human being, because we're all outbred, that the father's transplantation antigens would stir up an immune response in the female and cause her to reject the fetus. There's still argument going on about the extent to which fetal loss might be accounted for by this mechanism. To my mind, it's not terribly compelling, but there's no doubt that in certain circumstances, it could happen. But here is a circumstance where Anne looked at it. She said, okay, multiparous females who've had lots and lots of litters, what about the sex ratio of the later ones? It was known from my work and from work that had been done previously that there was a transplantation antigen that was expressed by males, but not females. So would you expect the mother to make an immune response to this male-specific antigen and selectively reject the male antigen? So the male's fetuses. This could be investigated by looking at sex ratios at birth. So this is what Anne did and found that there was no difference between them, the first litters and the last litters. So that it's very important to disprove hypotheses in science. You can't prove a hypothesis, you can only gather data that's consistent with it, supportive of it. Now, chimeras obviously were of interest to immunologists like me, but also to a postdoc, Takeshi Matsunaga, a very bright Japanese who'd done a PhD at the Basel Institute of Immunology and then had moved to London and worked in Anne's lab, and was fascinated by the idea of tolerance in these embryos. And we decided to do some experiments together, checking whether the cells, the lymphocytes from these were mixed lymphocytes, so they were from both different parental types, and whether they were tolerant of each other. We knew that those mice would accept skin grafts from each parent, but if you tested the cells in vitro using a new in vitro test that I had been working on, were they tolerant? And the answer is yes. And a third member here was this piece of work was Tommy Mayo. He, in fact, was the uncle of Francesco Colucci, who's the first author, the junior author, in fact, of a paper that was then subsequently published with Anne and I as co-authors, when very sadly, our friend Takeshi Matsunaga died prematurely in Sweden. But meanwhile, he had actually moved from Anne's lab to Susumu Ohno's lab in the City of Hope in California. And that forms another chapter, interaction with Susumu Ohno and his hypothesis, that I'm going to tell you about. However, before starting on that, I just wanted to tell you in a little aside about another enthusiastic, perhaps over-enthusiastic, Australian postdoc called Ted Steele, who bounced into London from Australia, actually from Canada. He'd gone from Australia to Canada and persuaded Reg Gorzinski to do some experiments with him, and then showed that claimed that they'd showed inheritance of acquired transplantation tolerance from males that were born of being injected at birth with Mediware's method of blood cells of a different strain of mouse, which were known themselves, those injected males, to be tolerant. Did they give birth to tolerant young? Was Lysenko at last going to be shown to have some evidence for his inheritance of acquired characteristics? Well, Mediware, who didn't actually think that this was the case, nevertheless generously agreed that Steele could come and work in our laboratories and repeat his experiments alongside us. And Anne very sensibly suggested that surely chimeric males, who themselves were tolerant, as we'd shown, their progeny should be investigated for tolerance. And these two papers describe different approaches our two labs took, which actually disproved Steele's hypothesis using this experimental method. It didn't please Ted Steele very much. He spent the rest of his time in London, mostly in Fleet Street, trying to get the interest of the red tops. Not entirely. And then he subsequently went back to Australia and caused anguish and trouble wherever he went. But perhaps that ought not to be quoted, because he will end up by suing me for defamation of character. And I don't think I want to appear in the High Court to defend that. Right. Now, Anne and I combined interest in her immunology, her skills, mine in transplantation, to explore Ohno's hypothesis. She wanted to, as I've mentioned, determine what determined embryonic choice between male and female. Was it a gene on the Y chromosome? And she described that it was probably a gene on the Y chromosome, and she named it TDY, tested determining Y chromosome gene. And meanwhile, I'd also developed ways independently, and not to address that question, but ways to measure immune responses of female mice rejecting skin grafts from males of the same strain. I mentioned that experimental setup a few minutes ago. And my aim became identification of the Y chromosome gene that was responsible for the expression of the HY antigen. And I wanted to find this out because this could be a route for inducing tolerance to grafts, organ and tissue grafts, to be used clinically. It was much more complicated than that, but I won't go into that now. Now, Ohno came along to suggest that these two were the same. because he said that as the Y chromosome was small and had very few genes, and therefore was assumed to have very few genes, that's not in fact the case, expression of the HY transplantation antigen in the embryo was itself the trigger for testis determination. So these two genes were one and the same. Now, in Anne's mouse colony, there were some males who carried a mutation called sex reversal, XYSxr, and their litters had a preponderance of males, including sterile males who had small testes and 2 X chromosomes. So they'd obviously inherited from their father something which switched them on to testis determination, despite having 2 X chromosomes. That particular inheritance pattern implies that XYSxr was on an autosome. But Anne had got another mutant in her mouse house, which had already started making her doubt about the autosomal location of this gene. But first, let me show you the results of our first experiments using just the straightforward, or relatively straightforward, sex reversal carrying male, SXY, plus this extra bit of XYSxr, which was either attached to the Y or an autosome. And when they were, I'm listing here the phenotype of the animals when born. So there were 3/4 of males born and only 25% females. The females were all XX. The males had 3 different karyotypes. 1/4 of them were XY. Another 1/4 of them were XYSxr like their fathers. So they were carriers. And the other remaining 25% had 2 X chromosomes and this XYSxr segment. However, finding that and here's the HY typing of them. So everything that was male was HY positive, and of the ones that were fertile, most of them were fertile except for the males that had 2 X chromosomes. But finding XXXXR males that are HY positive could be consistent with having HY and TD being with either 1 gene or 2 linked on the chromosome or an autosome. So we had to go back to the drawing board and use them. This is the paper that we published showing this result. Anne's other mutant was called T16H, which has actually been developed in Harwell by Bruce Katanach, another of Anne's long-term colleagues and friends. And she had potentially informative mice with this, she'd bred from these mice. And when T16X females were bred with fertile XYSxr males, the progeny included T16X females and males. So they both had the same karyotype, as well as XYSxr males. And the importance of this paper, which I won't go into the details of, but this key paper of Anne and Marilyn Monk in her lab showed that the chromosomal segment with genes causing sex reversal in XX mice was exchanged between the X and Y in carrier males during spermatogenesis. So you say, what's so special about that? Because up until that moment, it was thought that no exchange of genetic material could occur between X and Y, because otherwise it would mess up sex determination. But in fact, there's a very small amount of, there's a small area of both the X and the Y that need to exchange material in order for normal spermatogenesis to take place. And in Anne's lab, she and Paul Burgoyne worked further on that later. Using this particular breeding scheme, we wanted to, let me go back, I don't mess things up totally. We used this breeding scheme to see if we could find a recombinant mouse. That means a mouse that's inherited a broken up chromosome, which has separated 2 things that you're interested in. And we were looking to see if we could find a recombinant chromosome. These sorts of things, very often, depending on how far apart the genes are, if they're very close together, they're very rare occurrences. Now, this is it's a diagram of the Y chromosome of male mice. The bit at the end called PAR is the pseudo-autosomal region, and that's the bit that exchanges with an equivalent region at the tip of the X chromosome. The long arm has got other genes on it, which I don't know very much about, but I suspect others do. The YP, the short arm, is the one that actually is carrying the SXR. How it got there is another interesting story, but I won't press on with that. But just to tell you that that SXR was there, and our mouse that we have discovered, 1 out of 2,000 tested, had a deletion between 2 genes, ZFY1 and ZFY2, so that it clipped out all the genes between them. And those were all the HY genes. However, leaving Anne's gene, which has now been unfortunately renamed SRY, that is in fact her testis determining gene, still in place. So the mice continued to be sex reversed, but they lost the expression of HY. Fortunately, the mouse in which this was first discovered, this was the mouse in which we discovered this, was a female who was fertile. It's no good having an infertile male saying, oh, we've done this. You've got to be able to transmit this through the generations. And for reasons that have to do with X inactivation, it is possible to have males and females from females that are carrying this deletion. We called it the SXRB deletion interval. And the genes that are shown in red and blue underneath that is just our deconstruction of what those genes had in terms of stimulators of the immune response for skin graft rejection and generation of cytotoxic T cells. Now, this deletion mutation actually led not only to the identification molecularly of HY and also of the SRY, but also other Y chromosome genes that were involved in embryonic development. I've explained that the deletion was caused by a fusion event and that it separated HY from TDY, SRY. But then subsequently, it was very important to show that SRY really was the testis determining gene. And this was done by Peter Koopman and Robin Lovell Badge, both of whom had worked in Anne McLaren's lab. And they created transgenic mice by injecting just the SRY gene into XX eggs, fertilized eggs, and what then was born subsequently were XX males, sex-reversed males. So that showed that that was the functional gene for testis determination. And as I've already mentioned, Anne's colleague, Paul Burgoyne, continued to work on that particular section and find more spermatogenesis genes. So it's not true to say that there are few genes on the Y chromosome. It's packed with all sorts of fascinating genes, but I haven't time to tell you about those now. Let me now move, though, to looking at Anne's activities as herself a midwife. She's obviously a midwife to all of these mice that were born from manipulations that she'd designed and done in the lab. And that's the work I've summarized those experiments in the 3 lines at the top of this slide. But as I've already mentioned, those embryological research experiments were the things that underlay IVF development. And they were widely read and influential, and in fact led to the birth in 1978 of the first test tube baby, Louise Brown. That was experiments that were carried out by a laboratory scientist, a gynecologist, and a lab technician. The lab technician did all the manipulations in vitro and was never mentioned. I'm afraid I haven't got the names of Paul Edwards was one of the males involved here, but it's rather like the Rosalind Franklin story, that this technician who actually absorbed all the crucial information about how to handle the eggs, how to get them fertilized in vitro. She was not, her name is now known and is in the record, but at the time she wasn't fated along with the 2 men. fated, they were actually criticized very heavily for doing things, for messing around with nature. And this was not illegal because there was no act of parliament to make it illegal, but people thought it ought to be made illegal. And that public furore led to the setting up of the Warnock Committee by another Oxford figure, Baroness Warnock, who was a philosopher, but a woman of extraordinary powers of reasoning. And she and Anne had a very good interaction. Anne, being the only scientific member of this committee, guided them to decide under what conditions it was acceptable to do these sorts of experiments, these sorts of maneuvers to allow people who were infertile to have children. And one of the key things there, which is now, right now being talked about, is the limitation that if you grow human cells, sperm and egg, fertilized eggs in vitro, you can only keep the embryos alive in vitro to day 14. That was a point that had been identified by Anne in discussion with her experiments as being one that should be acceptable on a variety of ethical questions. That now, because there is the potential ability to keep them going for longer than that, that is now up for discussion. But the law in the Human Fertilization and Embryology Act, which was passed in 1990, is a real model for other countries for setting up a regulated system for IVF. And Anne herself sat on the as a member of the Human Fertilization and Embryology Committee as an authority, as a member for 11 years. As I mentioned before, she retired as the director of the Mammalian Development Unit in '92 and moved to the Gurdon Institute to pursue her research on germ cells. But that didn't occupy all her time, or she found enough time and energy to also serve as the Royal Society Foreign Secretary from '91 to '96, and also the vice president of the Royal Society. She was the first woman official of the Royal Society. Was much valued and much loved, in fact, by her fellow members. She was always very good at talking with people. She was awarded the Japan Prize in 2002, which is second only to the Nobel Prize. And here she is in Japan, having received the prize with two young women. Now, to finish, I just want to reiterate how good a communicator she was, because it was such an essential part of both her creative science, being able to interact with colleagues, research fellows and students and technicians, both in her own lab and in other labs when she visited them, and also on committees. She would sometimes be quite mischievous in very small groups of people on those committees, had a wonderful sense of humor. She also had a very strong social commitment to using science for the benefit of all members of society, including the poor, the disadvantaged, and those who were involuntarily unable to have children. On the fun side, she was a very lively host and guest for parties, dinner parties. Conversations during these events encompassed not only science, but also politics and much else. She was a wonderful friend to me. And during our years together, 75 to 2007, she came to my annual New Year's Eve parties, latterly with Donald. He'd moved back to London. She enthusiastically joined a witches' dinner each October the 31st, our trio being completed by Sohaila Rastan and Miranda Robertson. And I remember also dinners at her house in Steele's Road with a table full of very interesting luminaries, including Lewis Wolpert. So she was a multi-talented, much loved and very effective woman and scientist, and a model for other women to follow. and to finish, this rather nice photograph of Anne and Donald. Jonathan, thank you.